Imagine going into a hospital, right? You expect it to be sterile, a place for healing. But what if the bugs there, the superbugs we worry about, found a new trick? Like, actually eating the medical equipment.
It's a pretty startling idea, isn't it? We're talking about a discovery where a really common hospital bacterium, Pseudomonas aeruginosa, seems to have learned how to digest certain types of medical plastic.
Okay, wow. So this isn't just like a lab curiosity. This is potentially happening with things in people or used in hospitals.
Well, that's the implication. The specific plastic identified in the study, polycaprolactone or PCL, is used in things like dissolving sutures, stents, drug delivery patches.
Stuff lots of people might actually encounter.
Exactly. And Pseudomonas aeruginosa itself is a major player in hospital-acquired infections. It's already tough to treat because it resists a lot of antibiotics.
So this research, it came from Brunel University London, right? Professor Ronan McCarthy's team.
That's right, published in Cell Reports. They basically found this bacteria can, well, feed on PCL to survive.
Which kind of turns things on their head. I always assumed medical plastic was basically inert, that bacteria couldn't break it down.
That was the general understanding, yeah. Its stability is a big reason it's used. The idea that a bug can use it as essentially lunch changes the game. It raises questions about, well, device longevity and also infection control.
Okay, so you mentioned PCL specifically. That's the one they confirmed it eats.
Yes, polycaprolactone. It is designed to biodegrade eventually through chemical processes in the body over months or years.
Right, slow process.
But what the researchers found was different. It was bacterial digestion. They pinpointed the specific tool the bacteria uses, an enzyme they called PAP1.
An enzyme? Like a digestive juice, basically.
Sort of, yeah. A protein that breaks down the PCL. And get this, in their lab tests, this PAP1 enzyme chewed through almost 80% of a PCL sample in just 1 week.
1 week. 80%. That's fast.
It's remarkably fast compared to natural degradation. And the really striking part, the bacteria weren't just nibbling, they were using the breakdown products as their only source of carbon, their only food to stay alive.
Wow. So it's not just damaging the plastic, it's actively benefiting the bacteria.
And there's another layer to it. That breakdown process actually seems to help the Pseudomonas in another way.
How so?
Biofilms. You know those slimy protective layers bacteria build?
Oh yeah, the ones that make them super resistant to antibiotics in our immune system. Nasty stuff.
Exactly. The study found that the fragments created when the plastic breaks down actually help Pseudomonas aeruginosa form tougher, more robust biofilms.
So wait, by eating the plastic, it gets food and it builds a stronger shield against treatments.
That seems to be the case, yes. A double whammy, really. It makes an already dangerous bug potentially even more resilient in certain situations.
And Pseudomonas aeruginosa is already a big deal in hospitals, you said.
Oh, absolutely. It's a leading cause of hospital-acquired infections. Think ventilator-associated pneumonia, infections from catheters, often involving plastic devices. Plus, its antibiotic resistance puts it on the World Health Organization's critical priority list.
Yikes. So finding out it can eat the plastic in those devices, that has a whole new level of concern.
It certainly does. It suggests another way it might persist or cause problems.
Does this discovery make you wonder about other plastics too? Is it just PCL, or could other medical materials be vulnerable?
That's a really important question. The Brunel team did find indications, hints of similar enzymes in other pathogens. It suggests this plastic-eating ability might not be unique to Pseudomonas or limited to just PCL.
So what other materials are we talking about?
Well, think about things like polyethylene, terephthalate, PET used in some implants, or polyurethane that stuffs everywhere. Bone scaffolds, dental implants, catheters, wound dressings, even breast implants.
Okay, so potentially a much wider issue then, if other bugs have similar enzymes for those plastics.
It could significantly change how we view the long-term stability and safety of a huge range of medical devices. But, that needs more research. This study is the first step.
Right. Early days. But thinking about hospitals now, how could this plastic-eating ability affect outbreaks? Could it help the bacteria hang around longer?
It's a definite possibility. If the bacteria can use plastic surfaces, maybe on equipment or bed rails or whatever, as a sort of emergency food stash, it could potentially survive longer in the hospital environment.
Making it easier to spread.
Potentially, yes. Professor McCarthy suggested a couple of things. Maybe we need to start considering plastics that are harder for microbes to digest.
Makes sense.
And maybe, especially if a hospital is dealing with a persistent outbreak, they should consider screening the specific bacteria involved to see if it has these plastic-munching enzymes.
So change the materials and check the bugs. That sounds like it could lead to needing different infection control strategies, maybe different ways of cleaning or monitoring.
It might. Current monitoring often focuses just on finding the bacteria. Perhaps we need to also think about how they're interacting with the materials around them. But again, we need more studies to really understand how common this is and what the real-world impact is.
Okay, so let's recap the main points for everyone listening. We've got this common hospital superbug, Pseudomonas aeruginosa, that's been found to digest a medical plastic called PCL.
Right, using a specific enzyme, PAP1.
And this isn't just like a weird party trick. It helps the bacteria survive by giving it food, and it seems to help it build stronger antibiotic-resistant biofilms.
Which makes it potentially more dangerous.
And while the focus was PCL, there are hints other plastics and other bugs might be involved too. It basically means we might need to rethink how stable these medical materials really are inside the body or in a hospital setting.
Exactly. That core assumption that these plastics are just inert background material, that's being challenged. They might be active participants in these microbial battles.
It really makes you think, doesn't it? What does this mean for designing the next generation of medical implants or devices? Do we need bug-proof plastics now?
It adds a new design consideration for sure. Resistance to microbial degradation might become as important as biocompatibility.
And what about sterilization? Are current methods enough if bacteria can, hide out and snack on the plastic itself?
That's another area needing investigation. It just highlights how incredibly adaptable these microorganisms are. They find ways.
So the takeaway really is vigilance, isn't it?
Mm-hmm.
This is a reminder that the fight against superbugs is constant, and they keep evolving in ways we might not expect.
Absolutely. We need ongoing research, we need to stay alert, and we need to be ready to adapt our strategies, whether it's developing new materials, new cleaning methods, or new treatments based on discoveries like this. It's a complex, evolving challenge.